Welcome viewers. Today in this session we will be talking about RNA, the different types of RNA found in a living cell along with its functional role. Before we move into the different types of RNA encountered in a living cell, we must first know what is RNA. RNA, which stands for Ribonucleic Acid, is one of the most important nucleic acid found in the living cell. RNA is important because it plays a very important role in gene expression, and it also has a regulatory role which was discovered slowly with time by different researchers. The RNA, because of its varied secondary and tertiary structure which it can adapt, can perform a wide variety of functional roles. The enormous number of secondary and tertiary structures are greater in number than DNA, which helps RNA to perform a multifaceted role. Now let us move to the different types of RNA. The different types of RNA found inside a living cell, whether a prokaryotic cell or an eukaryotic cell, are of the following types. The RNA common to both prokaryotic and eukaryotic cell are mRNA, rRNA and tRNA, while RNA playing the regulatory role found exclusively or only in prokaryotes are antisense RNA, riboswitches and bacterial small RNA. While RNA performing a regulatory function and found inside an eukaryotic cell are miRNA, siRNA, snRNA, snORNA, etc. We will be discussing about all these kinds of RNA, and so let us now first move to the different kinds of RNA. The mRNA, rRNA and tRNA are both found in the prokaryotic cell, are transcribed from a DNA sequence. Now these DNA sequences are present in the nucleus of the cell, or in case of prokaryotic cell it is found in the nucleoid region, while the RNA found in the eukaryotes like pre-mRNA, small snRNA, then snORNA, then miRNA, siRNA, the location of these gene sequences are found in the DNA, and these two are located in the nucleus of the eukaryotic cell. Now some RNA, or rather some virus, they carry genetic information in the form of RNA and is known as RNA virus. Now let us move to the location of these different kinds of RNA molecules. mRNA are located in the nucleoid of prokaryotes, or they are produced from the nucleoid region of prokaryotes and they are moved forward to cytoplasm, while in eukaryotes mRNA is found exclusively in the nucleus region. The RNA, like rRNA, is found in the ribosomes. Ribosome is a cell organelle found in both prokaryotic and eukaryotic cell, while tRNA, common to both kind of cells, are found in the cytoplasm. The other kind of regulatory RNA like snRNA and snORNA in eukaryotes are found in the nucleus and nucleolar region respectively, while siRNA and miRNA, they are found in the cytoplasm. In case of prokaryotes, antisense RNA and all these kinds of RNA are found in the cytoplasm of the cell. Now let us move to the functional role of all these kinds of RNA. Now before we move into the functional role of these kind of RNA, we should first know what is central dogma of life or central dogma of molecular biology. Central dogma of molecular biology was first enunciated by Francis Crick in the year of 1958. He postulated that DNA, which serves as a genetic information carrier in both prokaryotic and eukaryotic system, first converts the information to a codon sequence carried in the form of mRNA. Thus, the mRNA, which stands for Messenger RNA, carries a series of codons, and according to which the proteins or the amino acids are added in a polypeptide sequence. Now, the phenomenon by which the DNA is converted to mRNA is known as transcription. While after mRNA is synthesized, it is through the process of translation that the protein is synthesized. The amino acid or the sequence of amino acid present in a polypeptide is governed by the sequence of codons present in the mRNA chain. So this constitutes or forms the central dogma of life, and we can see here how RNA molecule is important. It is due to the participation of mRNA, tRNA and rRNA, proteins are synthesized, and this proteins forms or plays a functional role or a structural role inside a living cell. So after knowing about the central dogma of life, we will move further on to the different sections of RNA. So we will first start with mRNA, common to both bacterial and eukaryotic cell, or prokaryotic and eukaryotic cell. What is mRNA? mRNA, which stands for Messenger RNA, is the genetic information carrier from DNA to protein, or it links up DNA and protein with each other. mRNA, if we look at the structure of mRNA, it has got three components. The first component is a 5' UTR. What is a 5' UTR? The mRNA consists of a polarity region, or it is polar in nature in the way that it has got a 5' end and a 3' end. The 5' end contains a phosphate group, while the 3' end contains a hydroxyl group. Near the 5' end, there is a region or sequence of bases which is known as 5' UTR. UTR stands for untranslated region, followed by a protein coding sequence region, which is again followed by a 3' UTR. Now this together constitutes the structure of mRNA. What is the function of 5' UTR? 5' UTR contains a sequence which helps in the binding of 16S rRNA, which is present in ribosome, and thus it helps in initiation of translation. Accordingly, it also helps in increasing the stability of mRNA, and also it helps in eukaryotic system in the removal of introns. Whereas the CDS or the protein coding sequence contains only the amino acids, which finally, or it contains the sequence according to which the amino acid is synthesized in the final polypeptide chain, which is again followed by a 3' UTR. 3' UTR again contains an untranslated region which is present at its 3' end. Now let us move to the other characteristic features which are found exclusively in eukaryotic mRNA and absent in prokaryotic mRNA. The first of that is the cap. A cap is present at the 5' end of mRNA. Now, since it is present at the 5' end, it helps to protect the 5' end of eukaryotic mRNA from exonuclease attack. The cap contains an inverted guanosine which is added to the mRNA by the help of a triphosphate linkage. Along with it is methylated at the 7 nitrogen position of guanine. So it is due to the presence of cap that mRNA is prevented from exonuclease attack, and it also increases the stability of mRNA, and it helps in the removal of introns. The next characteristic feature which is present in mRNA of eukaryotic system is it contains introns. Introns are intervening sequences found in the CDS which do not finally appear in the mature polypeptide chain. The introns are removed from pMRNA by the process of splicing. Again, in this process of splicing, there are small RNA molecules called small nuclear RNA which participates and helps in the removal of introns from exons and joins the exons together. The 3' UTR is followed in case of eukaryotic mRNA by a stretch of adenine residues at its 3' end known as poly-A tail. The poly-A tail helps in the transportation of mRNA from the nucleus to the cytoplasm in case of eukaryotic cell, and it also prevents the mRNA from 3' exonuclease attack and hence again increases the stability of mRNA. So in this way we can say that in case of eukaryotic cell, the mRNA is synthesized in form of pre-mRNA which is further processed through the addition of a cap, removal of introns and a poly-A tail addition to yield the final mRNA.
Ma'am, why does post-transcription and translation occur in prokaryotes and not in eukaryotes?
Now, in case of prokaryotes, as we have seen that it contains only a mRNA molecule which is made up of 3 components. It lacks a cap at its 5' end and it lacks a poly-A tail at its 3' end. Hence, it is more susceptible to the exonuclease attack found in the bacterial cell. That's why the short life of mRNA is highly short, or we can say it is highly unstable, and hence it can be easily degraded by the exonuclease. In order to prevent the rapid turnover of mRNA, the mRNA transcription or the transcription is coupled to translation. So as soon as the mRNA is synthesized, the translation initiates so that the protein is immediately prepared. So in this way, it prevents the attack from the exonuclease and it uses the mRNA or the information stored in the mRNA as quickly as it can to produce a polypeptide chain. rRNA, which stands for ribosomal RNA, is found in ribosomes only. In case of prokaryotic cells, ribosomes found are of 70S type. 70S type can further be divided into 50S and a smaller subunit called 30S. The 30S smaller subunit consists of 16S RNA and some proteins along with it, while the 50S subunit contains your 23S rRNA, 5S rRNA along with certain proteins. Now 16S rRNA is highly conserved in nature in bacteria and has long been used as a phylogenetic marker to compare the evolution between different bacterial groups. If we look at the structure of 16S rRNA, it is highly conserved. What is conserved? This means the position of the bases in the rRNA molecule is fixed and does not change. Because 16S rRNA performs a wide variety of functions during translation, or it helps in the binding with mRNA, it helps in the binding at the amino acid site and peptidyl site of ribosomes. It helps in the binding of elongation factor G during your translocation step of translation. It helps in bringing the two subunits, that is 50S and 30S subunit, together to form the initiation complex. So as a result, if one of the bases is changed, its structure will be changed, and accordingly this kind of different functions cannot be performed by 16S rRNA. As a result, it is highly conserved in nature. Let us now move to the eukaryotic ribosomes. Eukaryotic ribosomes are of ATS type. ATS can further be divided into 60S subunit and a 40S subunit. Now 60S subunit consists of 28S rRNA, 5S rRNA, 5.8S rRNA along with certain proteins, while the 40S smaller subunit mainly consists of 18S rRNA along with certain proteins. So all this rRNA helps in performing a wide variety of functions which helps them to perform the translation very efficiently. Now let us move to the other type, the third type, which is common to both, that is the tRNA. tRNA, which stands for transfer RNA, and it carries the amino acid to the ribosome during the translation process. The primary structure of tRNA is a linear sequence of nucleotides and whose total length is around 70 to 90 nucleotides long. The secondary structures arises by the folding of the primary structures into corresponding stem loop structure. The secondary structure of tRNA was first studied by Robert Holley and his team, for which they were awarded Nobel Prize in Physiology or Medicine. It is also known as clover leaf model as it assumes the shape of a leaf of a clover plant. Now let us move to the secondary structure of tRNA. Now how does the secondary structure of tRNA looks like? It forms four main stem base or stem loop region. The first stem loop region is known as the acceptor arm, and it forms or carries or accepts the amino acids through the CCA terminus, which is followed by a D loop. The D loop is named because of the presence of dihydrouridine base in it, which is followed by an anticodon loop, which base pairs with the mRNA, followed by a variable loop, which the variable means the length of the loop is variable from one tRNA to another, which is again followed by a T-Psi-C loop. Psi stands here again for pseudouridine and is one of the modified base found in tRNA. All of these loops have certain physiological function, and accordingly they have been named so. Now if we move further, the physiological function of tRNA is shown by the tertiary structure. The tertiary structure is formed by the interaction between the bases present in the D loop with the bases present in the variable loop and in the T-Psi-C loop. So accordingly, due to this interactions of bases between the loops, it folds or brings the loops together and bends the cloverleaf model into a L-shaped structure. Now due to this L-shaped structure, what is happening? The L-shaped structure, if we look at, will find that the anticodon loop and the acceptor arm are at the opposite ends of the L-shaped structure, and there is maximized base stacking in the L-shaped structure to promote hydrophobicity of the bonds. Now after looking at all the structures of mRNA, rRNA and tRNA, we must discuss about the functional role of modified nucleosides. Now certain of the bases in rRNA and tRNA are modified post-transcriptionally to form this modified nucleosides. Now what are modified nucleosides? Modified nucleosides are like when in the native condition, in normal condition rather, uridine, it gets modified to form ribothiamidine or dihydrouridine or pseudouridine. Similarly, we have cytosine, which can get methylated to form 5-methylcytosine or 3-methylcytosine. We have adenosine under normal condition, which gets modified to form inosine. Now all of these modified bases on a larger extent or to a greater extent occur in tRNA. So after knowing about the structures, we should know what is the function of the modified nucleosides in tRNA or rRNA. wherever they occur, whether it in tRNA or rRNA, they are known to confer increased stability to tRNA or rRNA. Accordingly, this is the first point. The second point is the modified base are recognized by certain proteins of the translational apparatus which helps in the process of translation. The third is it is also recognized by enzymes like amino acid tRNA synthetases, which adds amino acid to tRNA, and thus they help in the process of, or in the initiation of the process of translation. So accordingly, we can see the importance of modified nucleosides in tRNA or in rRNA wherever they occur. Let us now move to the different regulatory RNA found in prokaryotes and eukaryotic system. The first of such a kind is antisense RNA. As the name suggests, it has got a complementary, or it has got bases which are complementary to the bases present in mRNA, as a result of which it binds with mRNA and it inhibits the translation and thereby inhibits the production of protein. So this RNA are known as antisense and they play a regulatory role in gene expression. When they are synthesized according to the cell's physiological needs. The other of such a kind is riboswitch. A riboswitch is a regulatory RNA molecule which can exhibit different conformations. What are conformations? That is the structures. So since they can exist in more than one structure and the structures affect the transcription or the translation process. As a result, mRNA may not be synthesized or the mRNA may not be translated if such a structure is formed near the Shine-Del Carnot sequence. As a result, they are known as riboswitch. So when the translation is to be switched on, the stem loop structure or such a structure is not found near the Shine-Del Carnot sequence of mRNA. Now let us move to the other kinds of RNA, that is the bacterial small RNA found in prokaryotes, and they are induced under conditions of high osmotic stress, nutrient starvation or nutrient depletion, etc. Again, this kind of RNA binds with RNA polymerase and inhibits transcription. Now, if we move to the eukaryotes, we'll find such a kind where the RNA are playing in the regulatory role are of siRNA and miRNA. They have been recently discovered by Andrew Fire and Craig Mello while they were working with the nematode C. elegans. siRNA stands for short interfering RNA and miRNA stands for microRNAs. Both of these kind are short RNA which are 22 nucleotide long and they play an important role in regulating gene expression.
Ma'am, what kind of regulatory roles are played by si and miRNA and how does it help the organism?
Well, siRNA or miRNA are induced under certain conditions or certain physiological conditions. Now, what is happening is in the case of both siRNA and miRNA, the RNA length is very short, that is they are 22 nucleotides in length, but the mode of action differs. How does it differ? siRNA, with the help of a complex which is known as RNA-induced silencing complex, abbreviatedly it is known as RISC complex, it binds with the mRNA wherever it is similar or finds base complementary and then chops the mRNA. Whereas the miRNA, it binds to mRNA but where at the 3' untranslated region of mRNA. As a result, translation is inhibited. While in this case, when siRNA binds to mRNA, it chops off the mRNA, as a result of which translation is not there. So these kind of RNAs are produced by eukaryotic systems mainly, and for which they were first discovered in the nematode C. elegans, and for which Fire and Mello were awarded Nobel Prize in 2006. Since then, they have been used, or they are the potential markers which can be used for expression of genes, or rather for silencing of genes associated with cancer, associated with other certain pathological diseases. So in this way, we can regulate the onset of disease or their symptoms. We learned from this session that RNA plays a very important physiological role inside the living cell. It performs a wide range of functions from viruses to multicellular organisms. Like in viruses, it serves as the genetic material, while in prokaryotes, it can be found in the form of mRNA, tRNA, rRNA, playing central role in gene expression, or in the form of regulatory role in the kind of riboswitch, antisense RNA, bacterial small RNA. The symbol of such a kind is found in eukaryotic system. Now, all these structures of RNA helps the mRNA or the RNA to perform a multifaceted action or role inside the living cell.